from the Gladstone Institute at UC San Francisco to talk about WikiPathways.
All right, I'm going to talk about WikiPathways and how to change the world. Well, not really. That's just a title I had to come up with 9 months ago, so we'll see what I can do about that. You know, the Earth is something like 12,000 kilometers in diameter. And the pathways that you would actually be changing at WikiPathways are more on the order of tens of microns. So we're talking about a very small corner of the world. But you know, on another hand, all of these talks are about community intelligence and about changing the way people think about database management, and thinking about how individual scientists can participate in the collection and curation of biological knowledge. And so that's a slightly larger scale. The typical scientist is a couple meters tall, I'd say. So it's a slightly larger, small corner of the world. I apologize for this depiction of scientists, but if you Google scientist, that's what we're supposed to be doing, is Googling things. This is one of the first image hits that comes up. So we have some work to do on our public image, I think. All right, I like to begin with this early example of a curated pathway diagram. These are our energy metabolism pathways and amino acid biosynthesis pathways. It sort of looks complex, crammed onto this one slide, but this was actually the good old days when things were fairly simple and stable, right? This was distributed as a wall-size poster that you would hang up in your office and use as a reference, a stable reference for years, if not decades. I still have mine, actually. And that's just not how it works anymore. Pathways now come in all shapes and sizes. We have high-throughput assays that are feeding into interactomes, into condition-specific signaling pathways, into pathways for development, for disease and drug action. And pathways aren't just static images anymore, right? They can also be used as research tools for data visualization and for data analysis. Here, for example, is a typical pathway from WikiPathways. Like the textbook pathways, it includes proteins and metabolites. It includes reactions and complexes, and identifies subcellular locations, so it looks familiar to biologists. A key difference is that each of these rectangles is actually a data object that's connected to a database of standard identifiers, so that you can map from practically any sort of data set, map your data onto these images. Here, for example, is some differential expression data, upregulated and downregulated genes in yellow and in blue. And you know, something special happens when a biologist engages a visualization like this, right? The room grows quiet and their focus zooms into a particular area that they're interested in. Some things are highlighted, and a number of what-if scenarios start going through their head, right? They say, well, what if this cascade here could be regulated by increasing, this drug? And that's exactly what happens when we take statin drugs to reduce our cholesterol. And so what I'm doing here with some cheap PowerPoint tricks is trying to demonstrate the act of visualization, right? But the image is actually just sitting there. Scientist takes it in visually, and it's allowed then to associate with all the other subtle associations that we have up here, right? From conversations that we have with colleagues at conferences like this one, from prior observations, of which only a small fraction are captured in publications and in textbooks, I would argue. If we could parameterize all the things going on up here and throw it into a supercomputer, we could then just put in all known interactions, molecular concentrations, reaction rates, and just read out answers to our questions in concrete units of information. But we can't do that, at least not yet. And even though this is a conference for intelligent systems, I'm arguing with this that this is one of many reasons why it's still really important to include humans in data analysis. So going back to the basic unit of pathway analysis, the pathway diagram itself, Here's another example from WikiPathways. One question is, how are these actually constructed, right? How are these models constructed? This isn't derived from a single measurement, a single experiment, right? Rather, it's multiple types of data collected in multiple labs over years that's carefully curated and selected to highlight what's most interesting to researchers, and that's what's on this diagram. In this one, for example, it's cancer drug 5-FU and some of its byproducts that are released into the bloodstream, taken up by cancer cells, where they then interfere with key pathways for cell survival. And so, you know, this doesn't include all the possible interactions for each of these molecules, right? We wouldn't want it to. It would just be one of those hairballs if it did. This is a curated subset. But it also doesn't include all the things we haven't figured out yet or haven't figured out how to put into these terms. And so all we know for sure is that this model is incomplete, and that it's definitely going to change over time as we figure out more about what's most relevant to the action of this drug on cancer cells, for example. And this is exactly what we had in mind when we started WikiPathways. WikiPathways is a wiki, like Wikipedia, but what we did is we ripped out the text editing tools and put in our own pathway drawing tool so that users can come to this website, search for a pathway, and then they can click edit. And when you're in edit mode, you could add, for example, a new byproduct and show it taken up into cancer cells and have it interfering or triggering apoptosis, right? And then once you click save, this new information is then available to all your colleagues around the world in this pathway model. You can add literature references, supporting evidence, and the entire research community is your peer review group for this contribution that you've made here. They can decide to approve it, decide to undo it, or start a discussion around it. And when you edit pathways using the editor at WikiPathways, you're not just editing this static image on the website, you're simultaneously editing an XML file format in the background. And so this file format has some BioPax elements included into it. It's XML, so it can be exchanged and accessed programmatically for the developers in the audience. In addition to XML, we also have web service access to all the content at WikiPathways. We have embed code that you can paste into your own websites to have an interactive pathway widget with any number of pathways in it. And we're working on converting the content and representing it as linked data so that you can semantically integrate and query it. The most common use case right now, workflow of getting data out of WikiPathways, is through the XML format. So one thing we did is write an app for Cytoscape, where from within Cytoscape, which is a network visualization, network analysis tool, you can query WikiPathways and bring in, import in these XML files. And then, of course, you could import your data as from an Excel spreadsheet and define how you want your data to map to different color gradients on the pathway view. This is, in fact, how I made this diagram that I showed earlier. And this process is completely interactive and dynamic so that you can actually explore your data in the context of biological pathways visually. I still think that's a really powerful mechanism. And then, of course, in Cytoscape, you can also bring in all these other apps to do analyses like shortest paths, clustering, or overrepresentation analysis. So you can see how we're starting to feed into this. Here's another example of a virtuous cycle, right? So data is synthesized into these pathway models at WikiPathways. You can then bring in orthogonal data and analysis and visualization tools like Cytoscape and allow researchers to computationally and visually assess their data in the biological context and come up with new hypotheses and ideas, which then can be taken back into the lab to be tested at the bench or in silico to generate new data, which can then be synthesized into new pathways. So this brings us all the way back to WikiPathways, and we've found that this wiki model in particular, which is the topic of this session as far as a community intelligence approach to collecting this information, has been quite successful. We were collecting pathways as an internal team for a number of years prior to launching WikiPathways, and this was our growth curve for a number of pathways in human, just as an example, in blue, and a number of unique genes on those pathways. And then you can see in the years following the launch of WikiPathways, we entered a new phase of growth on both of these measurements. And then in this last year, it's really taking off. I think the next time I plot this, I'll have to use a logarithmic plot probably for the number of pathways, which is great. This is just quantity, right? Something that's difficult to quantify is the quality, but I can tell you that our internal curation team is just thrilled with the quality and with the improvement of content that we've seen come in through this mechanism. And I can talk about that more in Q&A. Another chart to show is number of users, which has steadily increased since our launch in 2008. And something that's unique, I think, and why our wiki is working is that we have this really high percentage of registered users actually contributing edits. It's average around 22%. And I think that's telling us that our tools are easy to use and they're not creating a barrier. And you don't have to register to use WikiPathways, so there's lots of people who come to view pathways, download pathways, and use them in the research. So we have page views, unique visitor stats, and these don't include access directly through Cytoscape or access through web services. So this WikiPathways for us is a pathway curation team, really started as an experiment, but we've been really happy with it and it's working out so well. So at this point I'm going to switch gears slightly there we go, and talk about a few lessons learned when I was invited to this session and learned what this session was about. I was thinking that this might be worthwhile. Maybe I'll poll the audience real quick. How many of you either run or are thinking about running a wiki, some sort of crowdsourcing or gaming system in the sciences? Oh, that's actually what I thought. That's great. So these next set of slides are definitely for you, but I think they're also generally useful just in terms of seeing how we think about the WikiPathways project and how we think about the changing culture around these community intelligence ideas. So the first lesson is to set early obtainable milestones. We went online in March of '07 with WikiPathways, and we immediately declared success because our internal team loved using the tool, and if no one else ever logged on, we would have still continued to use it just for our internal curation needs. And I think that's really important that the tool is immediately useful and that you're not jumping ahead to, oh, well, once we build it and 1,000 people are using it, it'll be great. You have to be able to survive that intermediate period. It took almost a year before we had someone use WikiPathways that we didn't know, right? We had probably 25 contributors at this point, but we knew them all until this guy showed up. And so it's a really important milestone, and we've now sort of adopted that as the first birth date of WikiPathways, January of '08. Despite the title of this talk, I would recommend not trying to change the world when you're first starting off with one of these types of projects. So you want to work with and not against the established models for database management. In our specific example with Pathways, we worked with the Reactome group, for example, and we host their human pathways at WikiPathways, so they still have their their own curation model, but they can kind of survey the crowdsourced community approach and handpick from that and feed that into their established internal model. You want to work with existing communities, right? You're not going to grow one from scratch, and so you want to identify the communities that are already out there, whether they're model organism database communities or whatnot, that are interested in using, interested in contributing and participating in a wiki. You want to make sure your tool works with existing tools and pipelines that are widely used. So in our case, this was going after the Cytoscape community and building that Cytoscape app so that folks who were already familiar with Cytoscape could just bring in our pathways and work with them in a familiar environment. And then finally, publishing and attribution models. We know this isn't going to change overnight, and so it's important to figure out whether it's figuring out motivations to make sure those work in the current attribution systems. We're working with Eric Schatt, who's launching a new journal called Open Network Biology, which I'm pretty excited about, which each publication will also include a submission of a network model. A subset of those will be appropriate for being curated as pathways. So we're working with him to feed curation through publishing, and I'm just starting conversations with folks at PLOS about figuring out how to get closer and closer to the point of submission for collecting pathway models and contributions, changes to pathway models. Then once you feel fairly settled and have these things figured out, then I'd say go ahead and try to change the world. Why not? So tweak established models, right? You want to be in position so that when the old models fail, you're there. When KEGG announced that they were going to throw up a paywall last summer, WikiPathways was there with up and running with web services that could be plugged into all the same tools that were previously using KEGG. At the poster sessions here, I saw half as many references to WikiPathways as I did KEGG, and that's completely new. That wasn't the case a year ago. You want to also grow and redefine communities, right? Well, we're turning data producers and data users into data curators. You want to change dogmatic perspectives. Instead of this idea of expert curators, think of everyone as a potential curator, right? If your database is so complicated that the typical researcher can't contribute to it, maybe it's not their fault. Maybe your schema is too complicated. Another topic I could go on and on about is open knowledge, and you know, new scientific findings shouldn't just be published. They should be as figures and as text. They should be captured in computational formats, immediately available and free, and open to active critique and active curation by the entire research community. New systems of attributions. I love this concept of nano publications. That fits in really nicely with contributions to wikis, where a number of small contributions add up to significant work. And this is sort of redefining what it means to be a productive scientist, or maybe extending what it means, right? There should be more than one measure of your productivity than just the impact factor of your publications. And then finally, new analysis pipelines. Take your unique model and connect with other crowdsourcing resources to demonstrate new pipelines of analyses and curation. And I'll just wrap up with one example of something we'd like to do with WikiPathways. Imagine while you're editing a pathway, say you've just added this carboxylase, and then the right-hand panel dynamically updates with interactions and annotations from other sites that are relevant to that protein that you've just added to your pathway. And you, for example, here are possible interactions that are 1, 2, or 3 steps away from the protein you just added, and you could just drag these in and bring in all the evidence and literature supporting references that come with it while you're curating. And then likewise, the reverse, as you add new interactions in the pathway and provide supporting evidence, that could feed back into these other resources that are feeding yet other resources. Okay, so I'd like to thank the team of developers that I work on WikiPathways, listed here. And I'm also involved in the National Resource for Network Biology. Part of our mission is to support the development and use of network biology tools and resources. Part of that includes helping folks develop, whether it's apps for Cytoscape or their own network biology tools. And so definitely let me know if you're interested in getting involved in Cytoscape development or WikiPathways development. We also participate in the annual Google Summer of Code, where Google pays students around the world to write code for open source projects. You can find out more about both of those programs at nrb.org. And I should also thank our contributors for WikiPathways. While that's running, I'm happy to take questions. It will stop. It doesn't go on forever. Yes?
One thing that our group does a lot with the NCI Nature Pathways and also other ones that we can download is run our own tools to statistically evaluate them. I was wondering if we and others put a bunch of pathways into WikiPathways. You said there's some XML file for each individual one. Is there a way to pull out all of the WikiPathways into a text file or something?
Yep, absolutely. We provide batch download of the entire set in a number of formats, including the most rich is our own XML format, but you can also get them in BioPax format, for example. And you can access it as a batch download, or you can access it programmatically through web services to get your hands on the live content at any time, say if you want to keep that up to date.
Our instructions for these are on the website?
Yep. You'll want to look for a link that says development in the left side panel, but we can talk about that more too.
Hi, Alex. Great talk and great idea. I mean, I think the idea is great. So I have a couple of questions. One is, have you noticed any controversies where people sort of change a term back and forth, and how do you handle it? Because there are such things in actual Wikipedia.
Yeah. We're still waiting to, we're eager to have that problem. We haven't run into it yet. We're asked that a lot, even before we launched, that would be a major issue of, say, 2 powerhouse labs wanting their stories, represented and battling over an interaction. Hasn't happened yet. One slight pressure release valve that we have in place that most pathway archives don't is that we're open to having multiple versions of, say, the apoptosis pathway. The canonical one is only so useful, so we'd love to capture the apoptosis pathway that's specific to this cell type, to these conditions, to this stage of development, whatever. And so we'll take on the burden of organizing and having it be navigation and browsable, but we don't put restrictions saying, well, if you want to edit apoptosis, it needs to all fit on one pathway. So I think that'll help.
Okay. And my second question is about benchmarking confidence. Basically, do you have evidence to show that this community-driven process actually improves the pathways?
Right. Like I said, it's sort of difficult to quantify. If you have some ideas, I'd be interested. Some of the things I've seen about comparing it to other resources has flaws. Our quality control includes things like, asking for literature references when you do add something new. But again, as an internal curation team, we haven't seen anything come in yet that's been vandalism or that's been nonsense, frankly. This is one advantage that we have to the Wikipedia approach, is that we don't have a bunch of 14-year-old boys who would think it's funny to vandalize a pathway. It's just not that funny, so they don't do it.
Okay, thank you for the
Yeah.
answers.